thank the organizers of this conference for giving me the opportunity to share some of our research work with the young students. I see a good number of young students sitting here in the hall. So, my topic of today's talk is the fertile synthesis of aza-phosphor-heterocycles from functionalized cycloaminolates and related amines. So, looking at the audience, the students are here. I will emphasize more in my talk on the type of heterocycles. Like if you are studying chemistry as one of your major subjects at the undergraduate level, you are already aware about the heterocyclic compounds, and we talk about the synthesis of that. So during last few years, we have done some research work on phosphorus heterocycles. As we know, whenever we talk about heterocycles, our undergraduate students, what do you think? A heterocycle is a compound having nitrogen, sulfur, oxygen mainly. Okay. So now I am talking about the phosphorus heterocycles. So when I say phosphorus heterocycles, earlier the phosphorus heterocycles were used to be in the inorganic chemistry. The phosphorgenes, the phosphobenzene was studied in inorganic chemistry, but now during the last couple of decades, the carbon phosphorus analogy. So there is a book, The Rise of New Domain by F. Marty, Phosphorus Carbon Heterocyclic Chemistry. So it has given the phosphorus heterocycles the equal status in the heterocyclic chemistry, the heterocyclic compounds, as nitrogen, sulfur or oxygen heterocycles. So the phosphorus heterocycles, for the benefit of the students, I would like to give a brief introduction. Depending on the size of ring, the phosphorus heterocycles can be categorized into different categories. If we start with the 3-membered ring with one phosphorus, the compounds are phosphidene, then unsaturated phosphidene, then 2 phosphorus, diphosphidane. So likewise the nomenclature goes on as is for the nitrogen heterocycle. Then 4-membered rings, these all heterocycles are available with us. They have been synthesized in the laboratory and their properties have been studied. So 4-membered ring with the phosphorus is phosphitane, which is saturated. If it has unsaturation, dihydrodiphosphate. And 5-membered heterocycles are important because we study the 5-membered heterocycles first, like pyrrole, purane. So here we have the 5-membered ring with phosphorus. If it is saturated, the compounds are phospholene. If one double bond is there, the compound is phospholine because of the unsaturation. And then this is similar to the pyrrole. So this is phosphol, analog of pyrrole. And then we have here this category of compound where apart from one phosphorus, there is another heteroatom X in the ring, the 5-membered ring. And these compounds are called heterophosphones because in addition to phosphorus, we have another heteroatom. Then 6-membered phosphorus heterocycle. So this is analog of benzene. So this is called phosphobenzene or phosphine. So here you can see this is lambda 3 phosphine, and here we have another category of compounds, lambda 5 phosphine. So because the phosphorus has resemblance with nitrogen and some difference also, the phosphorus forms PCl3 and PCl5. The nitrogen can form only 3 coordinated compounds. So similarly here, because of that property, the phosphines are of 2 categories, lambda 3, the 3 valency, and lambda 5. Similar to quinoline, the phospho analogue are called phosphinoline. So then we have, because of the lone pair available on the phosphorus, some other categories of phosphorus heterocycles, phosphine oxides and sulfide, dihydrophosphine, and then some other complexes where the lone pair available on this phosphorus, which is not available in the pyridine, similar to where the phosphorus has this lone pair which can be donated for the complexes, so sigma complexes are there. So then there are other phosphorus heterocycles also available which can come under the category of macrocyclic compounds, polycyclic compounds and spiroheterocycles. The phosphorus can be present at the ring junction of the two rings. And in spirocompound is a compound where two rings they are joined at one common atom. So why we are interested in phosphorus heterocycles? Because of their application in the phosphorus heterocycles in homogeneous catalysis, in the coordination chemistry, molecular materials and biochemistry. So phosphorus is a very essential material in our biological system. So we are interested in the phosphorus heterocycles. My interest is in the aromatic phosphorus heterocycles, and these two categories they come under the aromatic category. Here we see this is an analog of benzene, and the second one is the 5-membered heterocycle having aromatic ring. The phosphorus a member of aromatic ring. And the compounds what we have synthesized they come under these two categories, where a 5-membered ring having a phosphorus is annihilated with another ring. Here I have shown this as a circle. This circle can be a pyridine ring, pyrazine ring. It can be thiazolene, thiazole, quinoline, isoquinoline. A large number of other ring systems are annihilated to this 5-membered ring. A nitrogen and a phosphorus they are essential. Apart from that, some other heteroatom positions are also possible. Here, if there is a pyridine ring, we see that nitrogen is at the bridge head. And this is another category of azaphosphore we can call it because of the 5-membered ring. The name comes phosphore because of the presence of one nitrogen azaphosphore, and here this is annihilated to one benzene ring. So we see that a large number of phosphorus heterocycle categories are available. So my interest was the synthesis of phosphorus heterocycles. We started with a very simple method for the synthesis. Here, salt, iminium salt. We know the pyridine or any nitrogen heterocycle, because of the availability of lone pair, the nitrogen undergoes quaternization. So, if there is active hydrogen presently formed. So now if you have a CH2 group here, the reaction is cyclocondensation reaction. But if you have no substituent at this position, if R2 is hydrogen, then there is only a 4-membered chain. But this undergoes disproportionation and then undergoes 1,5 electrocyclization. If there are PG students sitting here, they have studied the pericyclic reaction. And in the pericyclic reaction, 1,5 electrocyclization, so undergoes the pericyclic reaction and this compound is formed. So these two compounds, they look similar, but there is a basic difference. Here there is one electron withdrawing substituent, ester group, at this 3 position, and here at this position we have alkyl substituent. In the second type of compound, we have the electron withdrawing substituent, the ester group, both at 1 position and at 3 position. So in such type of compounds, what is the difference? The difference is in their reactivity. You know, these are the aromatic compounds like olefin, quinoline, isoquinoline. They are aromatic. The double bonds are conjugated. But here in these compounds, they are aromatic, but there is a difference. They are 10 pi electron system, but there is a difference that this CP double bond is susceptible to cycloaddition reaction. So these are the reaction type of reaction can take place at this bond, but only when we have this electron withdrawing substrate at both ends. So these compounds undergo cycloaddition reaction. And these compounds, they do not grow facile during the reaction. So now I come to the mechanism part of the synthesis of these compounds. So like here, if you take alkyl substituents at 2 ends, here you have R1 an alkyl group and R2 an electron withdrawing substituent. There can be another substituent in the ring at any other position. So the cyclocondensation call it 4 plus 1 cyclocondensation rule because of the 4-membered chain reacting with the phosphorus, the 1-membered part, and these compounds are formed. The structures have been verified by the X-ray crystallography. So this route involves the, as I told you earlier, this halide intermediate, the initial formation of this halide, which reacts with CCl3 to form this dichlorophosphanylated halide, which undergoes cyclocondensation. So we could isolate this halide, the intermediate, and its structure. Structure was verified. There were some interesting features. Like you have one PCl2 group. When we have this PCl2 group, there are two PCl bonds, and here we see that the bond length is very much different. One bond is only 206 picometer and the second one is 270. So there is a great difference between the two bond lengths. Why the bond lengths are different? We have investigated it with computational chemistry. So I will come to it after 2-3 slides so that you can see how computational chemistry can help us in understanding the principles of synthetic chemistry. So here, like in this slide, we see there is one electron withdrawing substituent here. The reaction is initiated at this carbon and halide is a product, intermediate. If you don't have an electron withdrawing substituent here, if you have a phenyl ring that is not so much electron withdrawing, the reaction is initiated at this carbon, not at this carbon, and a different type of intermediate is formed. And this different type of intermediate can be identified by the phosphorus NMR spectroscopy or the carbon-13 NMR spectroscopy, which shows the presence of two PCl2 groups at the carbon. And here again the cyclocondensation reaction gives you the phosphoryl OPG. Then if you don't have a substituent at this position, here you have hydrogen, here you have hydrogen at the carbon adjacent to this nitrogen. So here you see that this intermediate halide disproportionate into another compound which are called allylides, because here you see this part is similar to an allyl, phosphoallyl group. Okay, so it again undergoes 1,5 electrocyclization and you get again a similar type of heterocycle having electron withdrawing substituent at the both sides of the phosphorus. Okay, so until here we have seen that if we have electron withdrawing substituent here, the reaction starts here, and if you don't have electron withdrawing substituent here, the reaction is initiated at this carbon. So why this difference in the reactivity? So how can you justify it? We investigated this type of preference of the reactivity, the site selectivity, because the PCl3 or the base is reacting at one of the two possible sites. In this case, this carbon, in this case, this carbon. Okay, so the two types of descriptors, and with the help of reactivity descriptors, we can calculate the the bond length, the two bond lengths. Normally, if there is a PCl2 group, one would expect that both bond lengths will be similar. So we have studied electronic effects like inductive effect, hyperconjugation, resonance effect. All of us have, okay, hyperconjugation we have studied. What is hyperconjugation? Hyperconjugation is the donation of electron into anti-bonding CH orbital. So if an electron is donated to anti-bonding orbital, the bond order decreases. It is called no bond resonance. Okay? So it gives the stability to carbonation, to free radicals. So those effects, what we have studied in our books theoretically, here we can justify the difference in the bond length by the computational calculation of those effects. How can we calculate it? Here you see that this is a pyridine ring. Pyridine ring is an aromatic ring. Aromatic ring has the delocalization of 5 electrons, and this carbon has a negative charge. And if this negative charge is delocalized into the pyridine ring, so what will happen? This nitrogen will remain positive and the negative charge can be delocalized from this carbon onto these 5 carbons here. But in such case, this carbon, the plane of this carbon and the plane of the pyridine ring, they will be in the same plane, so that the conjugation is effective. But when you search the database and find out the structure of the halides, you find some of the halides have such type of structure, but bond lengths are not equal. So what does it mean? That this 2 PCl bond lengths being not equal gives some extra stability to the molecule. Okay? So here we can find out the pyridine ring and the carbon, they are planar. The electric charge can delocalize into the pyridine ring. Still it is higher energy symmetry, but here the electric charge is not able to delocalize into the pyridine ring, but still it is the lower energy symmetry. So what is the reason? The transition states were also optimized, and in the transition state you can see that the higher energy symmetries are there. So here what we could calculate the hyperconjugation, anionic hyperconjugation. So this is something similar to hyperconjugation, but a little bit different. The donation of the electric charge into this negative electronegative atom. Okay, here this is PCl bond is there, so chlorine is electronegative. So because of the electronegativity of this chlorine, it takes the electron into the antibonding orbital. So this is the antibonding orbital. Those students who have seen the sigma and sigma star orbitals, they can identify that this shape, these 4 balls, blue, red, and red, they indicate sigma star orbital. If you can understand, what is the shape of sigma orbital? Normally we show sigma orbital like in the center, when we have 2 carbons, in the center we have one lobe having one charge, suppose plus, and on the 2 ends we have small lobe with minus. Okay, and if we draw sigma star orbital, it is something like that. In the center is the node, on one side is this plus minus and then plus minus. Okay, so here this is the orbital on the carbon. The extra electron on the carbon is in the pz orbital, and this pz orbital is in the same plane to the sigma star orbital, and it can donate, they overlap. You can see this antibonding orbital and here this orbital, they are overlapping. Since they are overlapping, the electronic charge is donated from this orbital to this orbital, and any donation of charge from one orbital to second orbital leads to delocalization. And this type of delocalization is known as hyperconjugation. And any type of delocalization, hyperconjugation, it leads to extra stability. And because of this hyperconjugation, the molecule having 2 different bond lengths, because here only one PCl bond can be in a plane of The second PCl bond is not in the plane where the electron can be transferred. So one bond is longer. Which one is longer? Which one is longer? The sigma star orbital has got electrons. So the anti-bonding orbital has electrons. The bond order will decrease. The bond length will increase. So because of the hyperconjugation, this PCl bond length is longer. So here you can see, because of the presence of this chlorine, this hyperconjugation is taking place. If you replace this chlorine by hydrogen, here, if you replace it by hydrogen and again calculate the molecular orbitals and visualize them, here you can see the orbital on the phosphorus and the orbital on the carbon, they are orthogonal to each other, perpendicular to each other, and when two orbitals are perpendicular to each other, the electron cannot come from one orbital to another orbital. So, here you can see that the extra stability can take place if there is a possibility of delocalization of this charge into these two groups. So if this electron is able to go into the antibonding orbital of this carbonyl group and this PCl bond, so this electron does not go for delocalization into the pyridine ring and the geometry is perpendicular. But if you replace one of the groups with hydrogen like here or like here, in both of these two geometries, now this negative electron is not able to be delocalized into these groups. Now it goes for stabilization into the pyridine ring, and you have these two types of geometries. Here the pyridine ring and the elliptic carbon plane, they are same, and here also they are same. If you add one hydrogen here, now the charge is not there. You can see the elliptic charge is not here because the carbon has 4 valencies. So here again this pyridine ring and this PCl2 bond length, you can see now these two PCl bond lengths are same. So the two PCl bond lengths, they become unequal because of the phenomenon of hyperconjugation. So then with the help of computational calculation, some more phenomena have been investigated. Here you can see 1,5 electrocyclization versus 1,5 proton shift. So these all are the topics of pericyclic chemistry. The students who have studied pericyclic chemistry, they can identify that how the intermediates are formed and the shifts can take place, and we can study all these mechanisms, all these intermediates with the help of computational chemistry. So these retrocycles are very interesting because they show wide variety of reactivity. Because of the presence of this hydrogen at the carbon adjacent to phosphorus, electrophilic substitution reactions can take place. Then, because of the presence of this CP double bond, which makes these compounds aromatic but not as aromatic as the carbon analog, 1,2 addition reactions take place. They can undergo 1,2 addition as well as 4+2 cycloaddition. Then because of the presence of lone pair of electron at this phosphorus, 1,4 cycloaddition, oxidation and coordination reactions can take place on these phosphorus atoms, and a variety of reactivity of these compounds can be investigated. So, how much time do I have? Over, I think. So, I will skip next slide because they deal with the computational calculations of the reaction mechanism. Here you can see with the help of calculations, we can investigate where this metal coordination takes place. So, the computational calculations can help us in understanding which intermediate will be formed and which product will be formed. You can calculate the energy of activation, you can calculate the energy of reaction to find out. So these are some more phosphorus retrocycles. They are interesting. Here you can see this phosphorus is in hexa-coordinate state. The phosphorus is at the brick head of 4 rings. So these interesting phosphorus retrocycles can be synthesized in the laboratory. So I acknowledge my research scholars. They are all my research scholars who have done the above research work, and the financial support of the DSC is acknowledged. Thank you.
explained by Nilima ma'am.
Any question in mind? Yeah, you can ask one or two quick questions.
You are very fortunate to have Nilima ma'am among us. If you are having any kind of query regarding this lecture, you may ask. Thank you so much, ma'am. Please give ma'am a round of applause. Our next speaker is Professor Naseem Ahmed. Sir has done his PhD from Aligarh Muslim University, and presently sir is working as Associate Professor in IIT Roorkee. I would like to request all of you to please welcome sir with a huge round of applause.
Ring. That's why it is called heterocycle. Otherwise, I am not talking cyclohexane type of ring. We are talking heterocyclic molecules. And these all are somewhere natural products associated molecules, which means one or other form is present in the nature itself. Means these are different products. I am just putting this one for last 3 years my research in this slide, and if you look in molecules, all have at least more than 25 different compounds, and each whatever shown here, that is one paper. I will discuss some of these papers here. First paper, if you look here, we are talking design and anti-proliferative activity against some conjugates. We are talking natural products like flavone and flavone conjugates with canaproject. Canaproject is a prostate cancer drug, and that widely we are going to hook with another natural product to get enhanced anti-proliferative activity, especially we are looking anti-cancerous properties. Look here. These are the design, how we have designed the molecule, because if you look at these molecules, you can go for aldol condensation reaction too. But I am not following the aldol condensation here. Classic condensation you are studying in the textbook, that is not followed here, because that chemistry is not working. We have to follow some other chemistry, and that's why we are going to get some other type of derivative here. This is also flavone containing compounds. Our aim is to put a chain. If you look here, I am showing 1, 2, 3, 4 carbon chain at 3 positions. That is our aim, to bring the chain in the molecules, and that will increase the lipophilicity of the molecule. That's why they can interact with the proteins, interact with the receptors of the, especially we are going to discuss the breast cancer, where they can interact with the receptor with the help of hydrophilic chains we have put here. Same time you are looking iodine 25, 125. What is the iodine atomic weight? Iodine atomic weight number. What is normal iodine? Many chemistry students is here. No. It is 127. Yes, iodine is 127. Means I am putting here 125. That is a radioiodine I am putting here. Means we are putting for the imaging of the breast cancer, how they are going to be imaged inside the cells, and that is done with the help of aspect or PET technology. That is, I labeled this one with the help of Bhava Atomic Research Institute. We label this molecule with 124, 5. And if you look at chemistry a little bit, I am giving here flavor. It is not going directly with the Clausius reaction. That's why you are following different paths to bring the change in the end. And because glycine only work for one carbon. After ketone, there should be one or hardly two carbons. If you are going to increase the chain of the carbonyl chain, it will not work. Chemistry is failed to work in that case. That's why if you look at the basis in the optimization reaction, we are doing so many trials to get the right product. In this one, we are trying different conditions, different ways, but we are not getting according to aldol condensation. Finally, we got a new condition, that is, you are getting 95% yield with the help of this. This is not based on Clausius condensation. Same way, we are looking different derivatives for the formation of flavone. Iodine leveling, again, you have to bring the iodine. That is not again easy. You have to break the bromine into the tin derivative. That tin derivative is finally quenched with two different types of iodine. One is normal iodine to get the cold product, that is called cold iodine product, and other is radioactive iodine level, that is a radio product. And that radio product is going to be used as a breast cancer. Characterization, if you look at characterization, we generally go for the NMR. NMR is the first technology, and techniques to characterize the compound here. Bruker's, that is going to be used in 500 megahertz machine here. And if you look at 500 megahertz machine, it is not going to resolve the spectrum. That's why the bottom line spectra is further expanded in the top spectra to know the exact whether the spectra is a first order spectra or second order spectra. And that basis, if you look at each peak is enlarged in hertz to find out what is the nature, how they are going to be formed or not. This is the NMR, proton NMR, you are studying. Similarly, carbon-13 NMR. Carbon-13, this is the conformation of your product. Yes, it is formed. In the same way, high-resolution mass spectroscopy, if you are talking, you are taking the malt, you are getting a single peak. There is no other impurity in the product. You are getting the molecular ion peak of the molecule. If you are going to as a flavone, same derivative, what is change? You are going to change with the nitrogen. Instead of oxygen, we are talking as a flavone. And in this derivative, look, we are going to do some same type of chemistry for originalized or optimized reaction condition. And again, we are going to level with the radioactive isotopes as well as cold chemistry here. And you are getting some biology. If you are looking, IC50 is reported here. This is right now in communication, this paper. MCF7, that is the breast cancer cell line we have used, and we are getting this value for the activity. MDA, that is another, these are two progesterone receptor positive negative cell lines we are talking here, and we are getting different value for these compounds. Characterization, if you look at the characterization again, we have characterized on the basis of NMR, proton NMR, carbon-13 NMR, and high resolution mass spectroscopy. You are getting these things. These are all, if you look at different derivatives, you will get different products in the same fashion. These are the radioactive, nitro, again nitro compounds, we are going for the biology, IC60 we have calculated in the case of this one. And similarly, we got the image from the molecular imaging division of the bar, and here I have not put the image of that ID25 level compound. But anyway, this is the again characterization related with this derivative. These all are one class of compound having flavone and as a flavone compound. Here we are looking different chemistry. Tano project, this moiety, look one moiety we are putting at the heterocycle where it contains one nitrogen, one oxygen, and that is right now in the market. That is you are using for prostate cancer drugs. Now we are going to break that molecule and hook with the natural products. And with different chain, N value we have given and different, you say the activity, it is shown here, compound 42, 41, we are getting some activity related with the anti-proliferative action of this molecule. Again, characterization is not easy. All spectra is second order spectra, and that's why you need higher resolution. And above, we are going to show again it as carbon 13 here. Mass. This is another that same topics we have put directly in the chain here instead of this another derivative we are looking for. Again, same study, and we are looking this with the help of Prince cyclization. Through Prince cyclization, this is a 6-member oxygen-containing heterocycle we are talking. This is heterocycle. It is going to be derivatized in another paper we have published in European Journal of Chemistry right now. That is, we are looking instead of tosylate, that ring is converted into a right molecule, and that we are going to click chemistry, and click chemistry we are going for synthesizing another derivatives. And if look, we have already synthesized 30-40 derivatives. Out of this, 3G and 4G is giving good activity against the cancer cell lines. And XRD is also taught in this one. Again, this chemistry, that is a well-known click chemistry, but to get the better result, we have optimized different bases and catalysts. According to that one, we are getting different products here. Characterization, if you are looking, all are well characterized with the help of proton NMR here. Carbon 13 NMR, if you look at, I generally assign the number where which carbon is going to be appeared. This is another word. Here, oral derivative you are looking, and that is published right now in Tetrahedral later, and we are looking some more diastromers of this product right now to get the derivatization, further derivatives of this compound. If you look at all these assigned numbers, it is going to talk about different position of your different protons and so on. Carbon 13. This is another, we are hoping for another derivatives looking tricyclic in a linear fashion, and we are looking 2 nitrogen containing and oxygen nitrogen containing. Further we are going to expand the cyano and amino containing compounds in the right side. Similarly, here we are going to expand the chemistry of these products, and characterization, look, all are well characterized. You are getting the NMR, carbon 30, carbon 30, carbon 30. This is another chemistry again based on, if you look, this is, we are looking at a big molecule for the anti-proliferative activity, hooking a chain of heterocycles. 1, 2, 3 heterocycles we are going to hook in this molecule to get the title compound. It is given name is used. That's why you can get. That is also characterized with the help of NMR. That is characterized with carbon 30. All are assigned. The fix is given for different numbers. This is published in 2013. Another type of turmeric-based molecule. If you look the spectrum, that is two oxygen-containing heterocycles we have synthesized, and reactions mechanism I am not going to talk here. Characterization, if you look all products, they are going to be well characterized in the different spectra. Carbon 30. This is another, if you look, this is another derivative. We have 3 heteroatoms in the same molecule, that is published in Heterocycle Japan 2012, and we are looking continuous simultaneous oxygen nitrogen and nitrogen nitrogen containing heterocycle 7-member rings that also here, and that is chemistry is well worked, and we assigned here, I didn't put the spectra, actually it is expanding lots of slides, but if you look, that is well characterized again. NMR is working. Similarly, this we have put simultaneous 3 rings containing nitrogen, nitrogen, 5, different pattern. If you are going to talk about the heterocycle, that pattern is going to define their activity. And 1 heterocycle, 2 nitrogen containing, 3 heterocycles in 5, 6 ring formation. We are looking this molecule that is also well characterized, and that is reported in different ways. That is another, if you look here, heterocycle, that is also published in 2012, different based on turmeric with the amino groups on the turmeric molecule. We have hooked 3-component, 1 reaction system, 1 part 3-component reaction to get the a big, or you can say 2 consecutive nitrogen, oxygen, 6 member ring, and you are getting this product. That is we have characterized with this process, and this is carbon 30. We are looking here. Finally, thanks to my students who worked hard to get so many products and publications. Similarly, IIT Roorkee, who provided facilities to run the experiments, and DST and Biyani, that is the funding agency, who gives money to work on these different projects, and BICON 15 to call me to present this work. Thank you.
Thank you, sir. Any query that the student having?
Yes. Yes.
So, complex so very molecule. I think very efficiently, very low, good yield, less time. So can you just elaborate how your compound can be used for application part?
Application.
Yeah, where we can use it.
We have given some examples for breast cancer research here, and we are also screening for antimicrobial activity of these compounds right now.
So while we are using as a anti-cancer or drugs, whether they are compatible with the cells or what?
Yeah, it's working. Problem is next generation we have to, because these all are PhD students are working. Once they publish 5-6 papers, they are in hurry to get the degree and go for abroad. That's why if you are going to give another students for the work in the same field to elaborate the work, they don't like it, because they are the chemistry student. Biology, for biology you need a biology collaboration. So that they can take the molecule, they trial in different ways. They go for the application for different parts. Okay? That is the problem. Otherwise, we have tested some key leads we have detected. This molecule is working. We have 45 key molecule detected. 5 is working, suppose. We have to focus on these 5 derivatives. That is not a still. And for that we need a collaboration. Somewhere biology people, somewhere good in these activities, those who are involved.
Have you checked the indigo also?
Yes. Yes.
Indigo. So maybe very good candidate for that.
Yes.
Some questions from your minds? I really looking forward to hear you. Anyone? Don't hesitate to ask. Because
Yes, at least NMR you are studying in the class. Proton NMR, you see, we have a not the first order spectra. What is the second order spectra? You can ask at least. All are giving the second order spectra.
No? Anyone?
Yes.
Yeah, thank you so much, sir, for your nice presentation. My question is that in your talk you told about the interaction of... Thank you, sir. Ladies and gentlemen, recently we have joined our eminent guest, Mr. Rakesh Sani, with us, scientist in charge, National Test House. I would like to request Manish Biyani sir to give a floral welcome to Rakesh Sani sir. Please give him a big round of applause. Thank you, sir. Now it's time for oral presentation. For this, I would like to invite on the dais Riho Hayama from Saitama University. Please welcome Riho Hayama.
BioProt is a high-functional tool and used to explore biological function of biomolecules and cells. This is a model of BioProt. For example, after attachment of BioProt into some virus, antigen, antibody, and some other things, the labeled material shows fluorescence, which may be useful to determine the biological activity of the particular biomolecule or cell.